Next we have Andrew Schork. Not yet. Actually, you've seen Andrew's picture on several slides, Naomi and a couple of other people as well. So Andrew is a postdoctoral researcher based at the Institute of Biological Psychiatry, St. Hans Hospital in Roskilde. And he works with Thomas Verger's group, but he was in Brisbane for a while, and we caught up with each other at the Queensland Brain Institute just a couple of weeks ago. He's part of the extended iPsych family, and really pleased, Andrew, that you could come out to Brisbane. We hope to see lots more of people like you and whatnot in Brisbane, and also in a reciprocal fashion that we can come and visit you more in St. Hans. But today, Andrew's going to talk a little bit about some cross-tray analysis based on iPsych data and share with us some of the genome-wide associations. Andrew, thanks very much.
Thank you, John, and thank you for inviting me to speak here. I feel a little out of place, but it's a real honor to speak to such a nice audience, and thank you for helping organize the trip to Australia. It was an amazing opportunity. I've been reading Naomi and Peter's papers for 10 years, and to have lunch with them and ask them questions and have them point out typos and stuff. It was really fun. Okay, so I'll introduce the iPsych data that I'm working with just very briefly. So if people aren't familiar with it, it's described in a really nice paper by Carson, and it's a case cohort design where it's sampled from a Danish birth cohort, and that's all individuals born in Denmark between 1981 and 2005. With a known mother alive and living in Denmark on their first birthday. So it's nearly 1.5 million people that is being sampled from. 60,000 subjects were sampled because they had a diagnosis of one of 6 major psychiatric conditions. And this was, at the time of sampling in 2012, essentially all of the cases of these disorders in this birth cohort. 30,000 were drawn randomly from the qualifying population, so about 2% of the people born in Denmark in this window were ascertained and genotyped. The diagnoses were obtained passively from national health registers that aggregate treatment, instance, discharge, diagnosis. So you go to specialty care psychiatric conditions, you receive some treatment, you're discharged, you get a diagnosis assigned with that instance, and then we kind of aggregate all of those instances for each individual. I'm highlighting a couple features here in red because they're kind of unique about the Danish data, and I'll bring them up later. But also, I think something that's really unique here is that the cases are representative patients presenting to the Danish healthcare system for their age, because they're essentially all of the patients at the time. And the controls are representative of the Danish population. I think this is because iPsych was designed by epidemiologists. And I think this is kind of actually an interesting and unique feature for a genetic study. So what this data looks like when we set it up and clean it for GWAS, this is kind of a messy slide, but each column of this figure, if it's black, it's a diagnosis. If it's white, it's not a diagnosis. So it's a pattern of diagnosis that are observed in some of these patients. We talked a lot about comorbidity, a lot about multiple diagnosis of individuals. But the study I'm looking at, it's 46,000 people that have at least one diagnosis, 20,000 people that have no diagnosis. Because of the age of the cohort, it's not equally distributed of diagnoses. We have more diagnoses for childhood, ADHD, autism, and then also affective disorder, and less for later onset disorders, bipolar and schizophrenia. A large proportion of the sample only has a single diagnosis up to this point in their life, but there's an appreciable amount, about 7,000, that have multiple diagnoses. So this is just the data that we're working with, and I just like to describe it because I think it might be useful for how you think about things downstream. So just as an example, when I'm talking about ADHD, for example, I'm talking about any person who has ever been diagnosed with ADHD, regardless of their other diagnosis. I'll make a special note if I'm going to talk about people who have only been diagnosed with ADHD. I'll call them ADHD exclusive. I just want to emphasize a couple pieces of intuition that I'm bringing to this project that are kind of developing. So there's going to be excuse me if there's a little lack of fluency in a couple of these slides here, but I just want to make a couple points, and that's what's the goal of a GWAS. So I'll quote Peter and Naomi's paper. The GWAS is an experimental design to detect associations, and the goal is to better understand the biology. So there's not a mention here about getting population unbiased estimates of effect sizes. It's my opinion that the basic goal is essentially to generate lists of biological functions, genes, variants, and processes with the hope of reconstructing disease etiology. It's not necessarily about getting population unbiased in kind of an epidemiology sense estimates. And so an approach that people have used is to maximize the probability that cases carry causal variants and that controls do not. And so you see different study designs that people have employed, and some of these might be ascertaining for extreme cases, ascertaining for super healthy controls, matching the presentation of cases to archetypical expectations, enriching for family history, et cetera. And I was at a conference in Copenhagen in WCPG, actually, when I met Thomas, and that got me involved in this whole process of coming here and doing a postdoc, where there was a talk, and someone in the audience asked a very well-known geneticist, aren't you worried that using healthy 100-year-olds as your controls in an Alzheimer's study might bias your effect sizes? And he said, no, I'm misquoting, he basically said, no, that's the point. We push the penetrance around to enable discovery. So when we think about how studies are designed for GWAS versus an epidemiology study, there may be some differences that might be interesting to think about. And I think because iPsych was designed by epidemiologists, very careful population control with represented cases, representative controls, that might give it a unique features. We can decide if those are interesting and how we use those. So the first thing I did was I wanted to know if, like other studies had reported, common Snip variance measured by Snip heritability was appreciable in this kind of different diagnostic scheme. So I estimated the Snip heritability for the 6 different iPsych diagnoses. And then I also wanted to ask if the Snip effects were shared broadly across the disorders or were kind of specific to disorders. So for every pair of disorders, let me just say, the green bars here are my estimates from iPsych, and then I took kind of published data, and I either re-estimated the genetic correlations, or I just pulled them from one of Naomi's papers here for the Snip heritability. So here I'm going to talk about the Snip genetic correlations, and we see that the Snip heritabilities are appreciable, and there's a large prevalence of genetic correlations, which have been shown by others. I do note there's some differences between iPsychs and previous reports, which I referred to in the talk I gave earlier this week as statistically ambiguous. And the reason I said that is because the individual estimates have large error bars. So when thinking about doing a statistical test on are these differences statistically different, none are strictly significant, and a few are kind of suggestive. And also Naomi had some has done some work where she's shown that there is some variability in these quantities when you kind of look across independent studies, and the variability that we're receiving here is more or less consistent with the variability in the very few independent reports of these quantities. However, some of these differences are expected due to differences in patient ascertainment, and I think Naomi touched on that a little bit in her earlier talk. And so when I gave this at the PGC Drugs to Discovery, I kind of just stopped here and I said Snip effects are prevalent, Snip effects are shared broadly, let's move on. But I just want to share some ideas here, and this is where I say this is something that I'm kind of thinking about, but I think it has some connections to the epidemiology aspects of the study, and so I just want to share some thoughts and progress here. In order to do this, I wanted to think why these differences might exist, and then when I thought about why some of these differences might exist, I realized I had to have a better understanding of how we estimate heritability on the liability scale, and so I went back. It was a good thing that I was investigating this when I was in Australia, because I could get citations from people who have been walking on this very carefully. And so I went all the way back to 1933, and I read a paper by Sewell Wright that looked at guinea pigs, and he noticed that most guinea pigs have 3 toes, but every once in a while you get kind of a vestigial toe, and if you breed these animals based on this vestigial toe, you can actually reconstitute a 4th toe. And he noted that the breeding results could not be accounted for on the basis of recessive nor dominant traits. There was some sort of latency for this extra toe in normal guinea pigs that didn't reach enough threshold to be expressed, and that the development of the 4th toe depends upon the inheritance of several factors. So you have a polygenic model. Non-genetic factors play an important role. It was etiology complex, and so this is actually the paper that introduced, I believe, the idea of a threshold model where this dichotomy cannot correspond to alternative phases of a single factor, but a physiological threshold in the character defined by many factors. And so I thought it was interesting to think about some of these models we use for estimating heritability in relation to guinea pig toes. But Falconer extended this to look at the inheritance of liability to certain diseases estimated from incidence among relatives. So basically, you have a prevalence of a disease in a general population, and you posit that that's caused by a normal distributed liability. If you look in relatives that are known to have some genetic similarity to cases, then you see an increased prevalence, then that means that there must be a difference in liability here that's correlated with genetics, and this allows you to get an estimate of heritability. So you're asking, is disease liability estimated via these modeling these prevalences in this framework higher in genetic relatives in cases than the general population? Naomi, Peter, others have extended this to the case of Snip heritability, where you measure genetic similarity rather than use it from relatives. This is kind of, where's my, oh, there we go. This is kind of a long way to say that some of these heritability models, they're kind of based on a model where you have you're assuming that you're studying a population and that your controls are representative of the control population and your cases are representative of the case population. And given the way that Jiwa samples are sometimes ascertained, this might not always be the case. And so I thought this is kind of just an interesting story to think about. So when I go back to the iPsych data and I consider how I estimated the heritability, I tried to do some permutations to how I estimated this heritability to see how these parameters affect the real world estimates. So here what I did is I mimicked Jiwa's intuition. So going into this, I said, I'm gonna do what all Jiwa's people do, I'm gonna screen my controls. So among our 30,000 people that are randomly sampled from the Danish population, they have a various pattern of diagnosis. So if I wanna estimate the Snip heritability for ADHD, I collect all the ADHD cases, and then among the controls, what do? Originally, I had censored them so that I only took people who had no other psychiatric disorder. So here I'm kind of sampling for extreme controls, and I estimated the Snip heritability in iPsych at these orange bars. As Naomi said, with some simulations, this can cause biases in heritability, and there's a debate about whether those are meaningfully large differences or not. So I went back and I said, okay, I should try to honor this population model, and I should try to take advantage of the iPsych population scheme. And I re-estimated the Snip heritability for each of these disorders, where instead of comparing ADHD cases to super healthy controls, I used the fact that we had random population controls. And consistent with Naomi's theory, we see a reduction in the heritability estimates that are small, but just a demonstration of how that might turn out in practice. Another actual advantage of the iPsych data is that it's embedded in a long history of epidemiological research on the rates and prevalences of different diseases, and so we can model better how our ascertainment scheme connects back to that ideal reference population. So again, Naomi and Valter Peyrut have a paper where they talked about what happens if you use true population controls, where you actually don't know the rates or diagnosis of any diseases, and showed then, well, you'll have some cases mixed in with your controls by chance that will underestimate your heritability, and they proposed a correction factor. In our case, our controls are not entirely unscreened. They have been exposed to the Danish national registers for as long as they've been alive, but in iPsych, that's only to their current age, and we have patients born between 1981 and 2005. So I think Believe Espen calls this conditioning on the future. So if I put someone in a control category, I'm assuming that they're a control now and they will be forever. And that might not be correct. So I wanted to try to integrate this framework into our heritability estimate by computing the expected number of 2 convert cases as a part of our controls, assigning each person a case percentage based on some really nice work by Karsten, which actually talks about the prevalence and occurrence of each of the iPsych phenotypes in the Danish population. And so so here's what I did. I took, for example, the cumulative prevalences for men and women in the dashed lines and the incident prevalences in the solid lines, and I said, consider a 20-year-old man with no diagnosis of affective disorder. Because he's healthy at 20, he's aged out of part of his risk, but we know from the population data in Denmark, he has some remaining risk. And this is where I'm not an epidemiologist, and so if I make mistakes, people should please correct me. And it's an attempt to try to integrate these different resources. So I defined his remaining risk in this way. I said the probability of developing affective disorder, given that he's healthy now, is the lifetime prevalence minus the prevalence before. And so I said if the lifetime prevalence in a male is 10%, 1% chance before, he's 9% of a case. Oops. And then I summed these up across in the iPsych random sample population and to try to get at a correction factor where I could say this is the proportion of people in our cases who I would expect to convert, and then I use Naomi's very beautifully worked out transformations. And so what this does is it bumps up the heritability again. So here we had the super controls went down when we did population controls. Now when I try to take into account that they're only partially screened controls for the single disorders, the heritabilities go back up, and it goes up kind of as a function of the heritability and also the rate of the misdiagnosis. So for something like affective disorder where there's this extra bump late in life, we see that this actually bumps the heritability up quite a bit. But for most of the other, these are kind of maybe academic differences, and it's not clear if they make a big difference in people in practice. But I put this example in here just to say that iPsych study design might be uniquely suited for characterizing potential advantages and pitfalls of estimating heritability in different contexts. Whether these advantages are purely academic or not, or can be leveraged in more practices context, something I'd like to learn more about. But I just think it's an interesting aspect of the data. And I just was speculating, maybe if you wanted to try to propose what it would be like to do genomic predictions in real world populations, you might want to integrate as much information about those real world populations better and see if some of these minor differences may actually carry through in important ways. Another example you could say is here's the estimate we estimate for ADHD and schizophrenia. It's higher than was previously reported. An interesting feature of our data is they're young. So here's our distribution of cases by age in black and controls in white. This is from Carson's data again. We see that there's a large prevalence of ADHD in youth, but a lot of schizophrenia is going to come on later. So many patients haven't yet been exposed to the possibility of converting from ADHD to schizophrenia. But there are some people here who are working on how to estimate these transition probabilities. And so I stole this slide without permission. I hope it was appropriate. But I think it could be interesting now to look at, Naomi mentioned the impact of diagnostic misclassification. You could try to integrate all of this data into some kind of a framework to try to get back at returning heritability estimates to that idealized population scale. I have no idea how to do this, but I think it's an interesting idea. Okay, so now I'll go back to the research that I have done, and that's that given there's a lot of genetic sharing and SNP effects, we wanted to look for SNPs that represented this sharing. So we defined a very simple phenotype, which is, are you a psychiatric patient or are you not a psychiatric patient? And so in iPsych, we had 46,000 cases and we had 20,000 controls. We estimated the heritability of this trait, which is about 18%, and this heritability here will be, the SNPs that contribute most to it will be SNPs that load on all the individual variants and covariance components. That's assuming they exist. So we did a genome-wide association study for this trait of are you an iPsych patient or are you not? And we found 4 genome-wide significant loci. So these are variants where one form is more frequent in the patient population than the diagnosis-free population after accounting for 1 million statistical tests. We did, for downstream analysis, we want a nice robust set or list of genes to try to do enrichment analysis, kind of what Peter described in his talk. And so we used an FDR cutoff to select another 50 loci. If some people in the GWAS community are not comfortable with FDR, and I don't really know why, but this corresponds to a p-value of 1 to the -5th, more or less. So the first thing I want to do is ask, are these SNPs actually plausibly nonspecific with respect to their effects? So here I'm showing odds ratios. In green is the odds ratio for our are you or are you not a psychiatric patient phenotype. And then each of the blue lines represents an odds ratio comparing the not psychiatric population to each individual case group. And we do it in 2 ways. We say all ADHD patients or the exclusive ADHD patients. And what we observe across these top loci is that the effect sizes are generally in the same direction, and the error bars are kind of overlapping, so the effects look to be of consistent magnitude across disorders. I call them plausibly nonspecific because a statistical test on this is a little tricky. So if we look at this one, we could say, okay, it looks like it has a odds rate. It looks like the ADHD cases are different from the controls, as are the affective, as are the anorexia, and are the autism. Bipolar and schizophrenia are difficult. They have really large error bars, and so now you have to pick which null hypothesis do you want to reject. Do you want to reject the hypothesis that it's not associated, or the fact that it's associated with 4 others? Would you think that that is more likely that it's associated with the others as well? Right, and then so we have a similar pattern of this across these other 46 suggestive loci, that they appear to be plausibly nonspecific. So we want to ask if these are replicable SNPs. There's not a lot of large cross-disorder GWAS with the same distribution of cases and controls as iPsych. When we do GWAS, we select for a single genetic background, Europeans. That means we exclude a number of individuals from the cohort that were genotypes. We have 7,000 individuals that have one of a different genetic background. So it's much less powerful, but we did a GWAS here using mixed effects models to account for the differences in ethnicity, and then we correlated the effect sizes between the two GWAS. The green ones here are the original genome-wide significant loci, and the yellow ones are suggestive. So the genome-wide significant loci have the same sign in this, and there's a strong correlation between the effect sizes in the two GWAS. We also then turned to a published GWAS of schizophrenia, and we correlated our cross-disorder effects with the effects in this large GWAS of schizophrenia, and the genome-wide significant loci were sign concordant, and the effect sizes across all the suggestive loci were strongly correlated. I emphasize this schizophrenia GWAS because if you remember, iPsych has very few schizophrenia cases. So under the assumption that these are cross-disorder loci, testing them for replication in schizophrenia seemed to give more credence to that effect, because if they were specific to one disease, we would think it would be one of the childhood disorders in iPsych. Okay, Peter also talked about functional annotation of loci and how do you do this. It's a complicated task. There's a lot of tools and it's a lot of bioinformatics, so we make a lot of correlational predictions. Doing the experiments to validate some of these is difficult, it takes time and money. But here, for example, we kind of zoom in around this locus and we see a genome-wide significant SNP, but it doesn't fall in a gene, but it's around some genes. So which of these genes would we want to add to our lists to throw into some biological process enrichment analysis? It's an open question. So what we did is we turned to a recently published paper, which is called LD score regression for specifically expressed genes. What this method does is it chunks the genome up into collections of SNPs that are associated with tissue-specific activity, and then it asks, among those different sets of SNPs, are any of them significantly enriched for heritability? So we partitioned this cross-disorder heritability with the phenotype defined here. We examined 53 sets of genes from GTEx, which were SNPs that were in or near brain genes specifically expressed in different tissues, and we used another set which was defined in the same way but a different collection of expression phenotypes, and we saw nothing significant in these gene expression sets. Then we turned to the roadmap chromatin marks. So this is defining sets of SNPs that aren't necessarily in or near the bodies of genes, but are in and near parts of the genome that show epigenetic marks of being open or active, perhaps as regulators. And in this context, we did see some things, some enrichments that were significant after accounting for all 600 of these tests. If we zoom in and look at what they are, they're fetal brain. So open and active, and chromatin that's open and active preferentially in the human fetal brain. This also replicates in the schizophrenia 108 loci, this fetal enrichment. This was published in the LD-Seg paper and has been reported previously. But again, I try to emphasize to emphasize the cross-order nature, consider the schizophrenia paper. We also iteratively dropped each of the individual diagnostic groups from our enrichment test to see if it was dependent on, for example, the large ADHD, the large affective disorder, or the large autism. And the enrichment in the fetal brain marks was consistent after removing any one of the diagnostic groups. So in order to select these loci, we turned to a collaboration with Dan Geschwin and his postdoc transitioning to Professor Hei-Jung Wang. And so we wanted to take advantage of the 3D structure of the genome. So DNA has a linear structure. It's also packed into 3D structure within cells. And regulation elements can be linearly distal but close in three-dimensional space. So they had a paper in Nature which uses a technique called high-C sequencing to actually map this interactions between, in 3D space, chromatin. And they showed that this can improve actually the identification of plausible candidate SNPs in that paper. So I'll walk you through what this looks like very quickly. So here we'll return to this one loci. We'll draw our plot here. We have a bunch of SNPs we have the top SNP here and some SNPs that are correlated with it. We subset down to a manageable set using a bioinformatics program called FindMap. And what this does is it iteratively proposes that each of the SNPs here is causal and then tests if it can generate the pattern of correlated effect sizes. And then it returns a list of SNPs that you need to capture potentially causal SNPs with 95% probability. It's just a way to subset down so that we can start to annotate SNPs to genes efficiently. And what we did is we checked if each of these credible SNPs, we looked at where they were in the genome, and then we used Dan's interaction map to ask what part of the genome is this SNP near in three-dimensional space. And then we said, if this SNP contacts a gene in three-dimensional space, we'll say that that's a candidate gene for this locus. So for example, here there's a gene. If we said this is the closest gene, we would have picked this gene and this gene. If we do closest gene in linear space, if we do closest gene according to this three-dimensional high C, then we would pick this gene and this gene here. And so we build a list of candidate genes according to this annotation procedure. I just picked this one because I thought it was interesting. It's a calcium-mediated messenger that's involved in voltage-gated calcium channels, among other things. And Annika told me this after the Drugs to Discovery talk, but 2 weeks ago, Lundbeck announced they're doing a phase 1 trial on a PD-1E inhibitor to treat cognitive symptoms within schizophrenia. And so I thought that's an interesting piece of independent information. Of course, we haven't functionally validated in a biological sense this connection. It's a bioinformatic prediction. So among these 50 loci, we collected a candidate list of 280 genes, and then we asked what functions are these genes enriched for. This is my last slide. And what we saw is that they were enriched for fetal brain expression, which peaked around mid-gestation. So here we use a data set called BrainSpan, and we look at the average expression of these genes across time in human fetal brain, in human brain tissue, and we see that the expression peaks here around 20 weeks prenatally and is flat postnatally. This just shows the mean expression in the prenatal and postnatal period. Then we also took publicly available data which looked at single-cell RNA sequencing in cells derived from developing human fetal brain. And this enrichment here is for this gene list against list of genes that are most representative of the expression pattern in different populations of cell and human fetal cortex. And we identified radial glia and interneurons as two cell types that might be enriched for these effects. And then we also did some gene ontology categories, and we saw they were enriched for axon guidance and neural developmental function, among some other functions. And right, so we observed broad sharing. Maybe we can do interesting things with that. We identified a few SNPs that appear nonspecific. They were strongest in fetal brain regulatory DNA. We annotated them using regulatory maps developed in human fetal tissue, and these seemed to also produce a list of genes that had kind of convergent evidence in their functions as well. So the kind of broad hypothesis that we have tried to generate from this data is that common polygenic SNP effects that disrupt or alter gene expression during early fetal brain development may induce non-specific liability to later psychiatric outcomes. That's it. Thank you. Oh, I guess I probably need to make
Andrew, that's a very impressive body of work. And I must say that my heart leapt with joy when I saw Carsten's paper was influencing your analyses. That's our dream, isn't it? We want that. That was Carsten's Atlas paper. Fantastic. But so many other things were good as well. Questions for Andrew? Rome at the front.
So the FDR actually has a long history of being applied in genetics, but the overall conclusion is that it can't learn from the data because there's too much signal, too much polygenic signal. So that's why family-wise error rate approaches are favored, right? So it's hard to calibrate the FDR in a situation where LD is variable across the genome and things. So there's a good post on, don't ask me because I'm not an expert, but there's a good post on Genomes Unzipped by Mark Daly about why it's not favored in the polygenic context.
Yeah, the only defense I would have is that choosing a p-value threshold can be arbitrary as well. So do I want to do 5 to the minus fifth, 1 to the minus fifth, 1 to the minus sixth, 5 to the minus, and you see papers that publish at different thresholds. And so this at least lets the data tell me a threshold, so I can claim that I didn't look at too many different thresholds. And I did do a lot of simulations and doing things like LD pruning, where I pruned SNPs at all various different thresholds and looked at different minor allele frequencies, and estimating the FDR in about probably 20 different subsets of SNPs, they all converge on more or less 1 to the minus fifth as a p-value threshold.
Okay,
So.
well, I'll talk to you after.
Yeah, sure.
After. So, and then just the other question though is, so you did a cross-disorder GWAS. So one of the things is that people have only traveled part of the way through the liability distribution for some of the disorders, and that's variable. So one question that maybe relates back to the previous talk is, what's happening with the people with really high, let's say, schizophrenia polygenic risk score right now? How enriched are they for the childhood disorders? How enriched are they for depression? As say the top decile, how many of them actually have a psychiatric disorder at the current time versus what you would expect from others?
Yeah. So
any psychiatric
I guess there's
disorder.
two answers to that question. One is it's a great question for John. I think what comes before what and how many different things and the rates of diseases at different times. The other question I think that the point you may be getting at is, okay, if we have people who are young and they have polygenic risk, they might not have anything now, but they should have something later.
They might have another psychiatric.
Or they might have another. Yeah. So the conversion is something that I would love to talk to John about how to model that. It's a complicated modeling problem. The people who have no psychiatric condition and then will convert, in the context of our study, they'll bias us towards not finding anything. So from the GWAS perspective there, that should reduce our power to find something. The conversion rate, things that fail to convert, if it's an appreciably large rate, and that's probably a good question to think about with Naomi, is at what rate of conversion does this substantially change the pattern? That could make SNPs appear more, you have this, it's a diagnostic misclassification problem, like Naomi said, where you have a person now who's ADHD, but maybe later they'll be schizophrenia. So that means some of our ADHD patients will convert to schizophrenia. Then if you're a psychiatrist, you can decide about how you want to treat that earlier ADHD diagnosis.
Yes, as Andrew said, in iPsych it's 80,000, but in Oligard's data set there's 5 million people, so 84 million person years. So we can answer the question like what the probability is if you have this disorder going on to get that disorder at that level. And we hope that those estimates can train or inform, Bayesian sense, some of the work that Andrew's doing in cross-train.
And it is, just to make one more point, it is one reason why we went with what could appear like a very crude design, which is just lumping patients into a group and doing a case control, as opposed to one that would try to fit a model, which would identify, is this a 3 disorder SNP or 4 disorder SNP or 5 disorder SNP? So that approach has been used by others, and I intentionally didn't use that model because you have this problem with the diagnostic overlap. So we're kind of saying if they're general, they should be there in everybody. It's hard, some of these things are hard. It's hard to know if they're academic differences or how practical they are for our purposes. But it's one reason why we couldn't use some of these more specific kind of model selection approaches for our GWAS. One reason why I avoided them.
Andrew, thank you very much.